Hi, everybody. Sorry, I'm a couple of minutes late. All right. Let us continue. Questions from where we were last time. Anything about LTE? Okay. All right. So one of the observations last time from the experiment, this was work done about 25 years after the experiment had started. So this is just after 2010 or so. Was this question that as the cells are specializing on glucose in this case, and of course one of the lines is also specializing on uh, citrate, but if we ignore that part, essentially the cells are specializing on utilizing glucose. What is happening to the metabolic capabilities with regard to consumption of carbon sources which are other than glucose? Uh, what do you think? So if I tested the performance of these evolved lines on carbon sources other than glucose, how do you think that performance would compare against the ancestral performance in the same carbon source? So let's say arabinose, for instance. Arabinose is a pentose. uh, we compare the ability of the ancestor to grow on arabinose and the ability of the evolved strain after 50,000 generations to grow on arabinose. What do you think is going to be the outcome? Is the question clear? Yes, no, doesn't matter.
No, sir.
So the question is that let's say that we are talking about a carbon source other than glucose. Let's say it's arabinose. The ancestor will have a certain fitness in arabinose. Now we've evolved the ancestor in a glucose environment for 50,000 generations, and at the end of these 50,000 generations, what do you think is going to be the evolved line's performance in arabinose compared to the ancestor? Will the fitness in arabinose increase, decrease, stay the same? And what is our expectation with regard to the evolved line's performance in another carbon source? Does that make sense?
Yes, sir.
So what do you guys think? Anybody?
The fitness will be lower.
Okay. Why would you say that?
Because the individuals in the after 50,000 generations have never seen arabinose and they might have lost the genes to utilize it.
Okay. And why would they lose the genes to utilize it?
Because they would have become more evolved to utilize glucose and citrate, things like that.
That is correct. So, okay, let me come back to that. Any other thoughts?
If we continue growing them in the arabinose medium, they may become like the fitness may increase.
That's not the question, right? The question is that having been exposed to glucose and not to arabinose for the duration of 50,000 generations, what happens to the ability to grow on arabinose? Any other thoughts?
Maybe there's no selection pressure when mutations arise in those genes uh, which help arabinose to be utilized. right?
Mm-hmm.
So ideally, I think I'd also say that uh, uh, the utilization of arabinose might decrease.
Okay.
Yeah.
Okay, sure. Let me also again come back to that. It's the same comment as Pavitra. Sure. Akshat, you was trying to say something?
I was saying if it might depend on how
I'm
much
sorry.
of
Say that again.
yeah, I was saying it might depend on how much of the pathway they have in common.
How much
If
of what?
the sugar utilization pathway of glucose and arabinoids are largely common,
Hmm.
one might expect that growth in arabinoids might also increase in the evolved line, but
Hmm.
the pathways are not largely common. There's no
Understand.
pressure on those arabinoids utilization genes to improve or
Right.
or
And I
no
understand.
pressure.
Right. So everybody, and especially Pavitra and Anirudh, what do you guys say to that? What Akshat is saying is that if the two carbon sources, the utilization pathway is largely the same, then there is also an expectation that growth on glucose might actually enhance growth on arabinose too. And if the carbon sources are very, very different, that their metabolic pathways do not overlap, then sure, it's possible that uh, the growth fitness might decrease. But if the pathways have overlap to a fair degree, then the growth might actually improve. What do you guys say?
Yes, sir.
Yeah, it could be right.
Hmm.
I don't know what the pathways are, so I just told a more generic
No,
answer.
no, that's okay. That's okay. So I mean, in this instance, arabinose and glucose, there's a fair degree of overlap between the two. Um, so yeah, okay. So that is fair, and in fact, what is largely seen is that if you evolve an organism on a particular carbon source, maybe not for we don't have very many lessons to learn from about what happens when we evolve for a duration as long as 50,000, because there is no other example from which uh, we can understand this part of physiology. uh, but in shorter experiments, there's a large number of short-term experiments which have been performed, and what's generally seen is that evolution on one carbon source actually leads to enhancement of fitness on other carbon sources too. And that is largely to do with the fact that there is considerable overlap of how these sugars are performed. And again, we don't have time to get into these things. And I think we did discuss this once, uh, that the concept of there being in biochemical pathways, the concept of there being one rate limiting step, and that rate limiting step limiting growth, is a fallacy. It does not happen like that. What happens is, of course, there is a slowest reaction, but if you analyze the biochemical pathway, uh, it's fairly straightforward to show that even though one enzyme might be the slowest in an entire pathway, it's not as if growth is limited by that reaction only. Of course, if you improve that particular reaction in a pathway, growth improves to the largest degree, but it's not as if you improve the others, growth will not improve. If you improve other enzymes performance, growth also improves to a lesser degree, but the overall contribution of fitness is spread across multiple enzymes and is not just limited to the slowest enzyme in the chain. And this is a body of literature that was developed in the 1980s and 90s, And the distribution of effect of fitness is something called a control coefficient, and that is spread across a pathway. So when the cell is improving growth upon a particular substrate, it acquires mutations in its pathway, and there is a great chance that it actually ends up improving performance in other carbon sources because of that reason too. In the other argument that that growth actually becomes worse in arabinose as compared to the ancestor, there could be two reasons why this happens. The first one is that growth on glucose, if you enhance that, and I'm using arabinose as a variable here, so the study that we're going to be discussing actually they tested it for like 24 different carbon sources. The first argument why the evolved strain might become worse on an alternate carbon source, and I say arabinose here, is that there is a trade-off involved between utilization of these two sugars. You cannot improve at utilizing both glucose and arabinose. If you make one better, automatically, because of the trade-off involved, you become worse off in the other. So this has an adaptive reason for why you are becoming worse off on using arabinose. And that adaptive reason is that the fact that you are becoming better at utilizing glucose, you become worse at utilizing arabinose, right? And there is a mechanistic reason for that, and hence you can't improve at both. And the fact that experiment is done in glucose, you are getting better at utilizing glucose, and hence automatically you are becoming worse off at using arabinose. So this is an adaptive reason for you becoming worse off when growing on arabinose. The other reason is not adaptive. The other reason is more because of drift. And this reason is that actually you didn't really need the arabinose pathway. So even if you picked up mutations in the, arabinose pathway, which made these enzymes non-functional or less active than the other, then you wouldn't feel any effect of that mutation when the experiment was happening in this glucose environment. And then when you and this was purely neutral, the cell didn't really care. A mutation happened and it didn't care, and hence there was no adaptive value associated with that mutation. But when you transfer this particular genotype in an arabinose environment, now these mutations have an effect, and hence you see a decrease in the fitness of the evolved strain in this arabinose environment as compared to the ancestor in the same arabinose environment. Right? So this reason is a non-adaptive reason for seeing this decrease in fitness in this new environment. And around 2010, there were these two reports published, and one of them said that, so they tested performance of these evolved lines, all 12 of them, on 24 different carbon sources other than glucose. And when they saw a decrease in performance, and in some cases there is enhancement in performance also, but remember this is 50,000 generations, so a long time has happened. In one study, the argument, what they saw led them to conclude that this decrease in fitness in alternate carbon sources happened because of trade-offs involved in utilization of glucose and the alternate source, and hence as you were becoming better at glucose, you were becoming worse off at these other carbon sources. In the other study, which was published a couple of years later, they saw data which led them to conclude that actually you become worse off on other carbon sources purely because of drift, and it has nothing to do with trade-offs involved between these two carbon utilization pathways. So we will discuss that on Wednesday. But I just wanted to lay this context for you, and I'll share the two references. Just skim through them, and you have to comment on why you think the two studies led to these completely opposite uh, conclusions. All right. So I want to finish the discussion on citrate utilization. This all right. Okay. So the first question they asked when they saw citrate utilization evolve in these lines was the fact that did this happen via a one very rare mutation, or did this happen via accumulation of certain mutations over an elongated period of time, and only because historically this particular line had accumulated all these mutations did the final mutation take place which allowed these strains to utilize citrate. So this was part of what was there in the reference I shared. Does anybody want to mention how did they check this? Give me 1 minute. I just want to shoo away this crow that's there. Yes, Anirudh.
They hypothesized that if it was due to one mutation, then the probability of finding a citrate utilizing species would be the same throughout all generations. But what they found out was most of the citrate utilizing mutations, species, came about only after a certain number of generations.
Right.
The probability of finding them is much higher on later generations.
Right.
So they assume that it's probably because of the accumulation of other mutations that led to this.
Correct. Exactly. Right. So these are the two competing hypotheses, and the first one is a rare mutation. So and we know that all SNPs have taken place in all lines many times over. And this could happen if the mutation that facilitated this change, and the change being CYT minus to CYT plus phenotype conversion, this must have been via a very rare mutation, a very special mutation. And if it's one very special mutation, then that mutation could happen to any line at any point in time. Whereas the other one is called the reason of historical contingency, which is that it happened in this particular line because this particular line has gone through an evolutionary pathway which gave it the necessary mutations such that now it could acquire one more mutation and hence become capable of utilizing citrate. So geometrically you can think of it like this, that this was the starting point and this is the glucose fitness peak, and all these lines have taken similar but unique paths which are leading them to the glucose peak. However, this one line which is also improving on glucose actually comes at this ridge and takes this mutation, and this now becomes what these guys call potentiating mutation. And now in this background more mutations can occur such that you move towards this citrate peak. So So this potentiating mutation allows this movement, and for these mutations now, for these lines, going back to this citrate peak is now not possible because natural selection will not permit it, and it happened in this line at just the right moment. So to test between these two possibilities, of course this is a hypothesis. To test this hypothesis, what they are going to do is, if they evolve each one of these lines at this particular time point, so assuming at one particular time they were all at this level, so they take this particular genotype, and so on and so forth, and they evolve them in glucose. And what they are looking at is that if I evolve this particular line in glucose for a certain number of generations, how many cells acquire the ability to utilize citrate? Right? So I evolve this for 5,000 generations, and I check whether the evolved lines can grow on citrate or not. How does one check if what is a very straightforward way of checking if individuals in a culture can grow on citrate or not? Anybody?
You take a plate with citrate and on the original culture, we just stick it and see which colonies are growing on citrate.
Yes, absolutely. So that's exactly what they did. So so you grow cells up, and at whatever point you want to check if there are any individuals there which are capable of utilizing citrate, you just take all those cells and spread it on a plate where citrate is the only carbon source. And if there is one or many individuals in that column, which in that culture which had citrate users, then those would grow to become colonies. Otherwise, the rest of them wouldn't grow, and they would just simply die on the plate. And that's what these guys did. That they evolved each of these lines from several starting points, and they're looking that how many cells evolved to grow to utilize citrate when they are evolved from various starting points. And now the argument is that if it is a rare mutation hypothesis, if it was one single rare mutation that happened that facilitated the cells from you to use citrate, then that rare mutation is equally likely to happen in this line as compared to this line. So when I do this evolution experiment, the probability of arriving at that citrate user is equal in each one of these lines, and that's what I should do. That's what I should see. So that's the hypothesis. On the other hand, if it was this historical contingency argument, then this is already gone on a path from where going to this particular direction is impossible, and hence I would only see citrate users evolve on this particular line, and the probability of citrate users noticing citrate users increases as if I were to take this as the starting point. Now, if I evolve cells from this as a starting point, again, it could go anywhere. Some of them could go here also, but there is a small probability of this. Now, if I evolve them from here, then the probability of evolving citrate is higher, but some of them could still go here. Whereas at the closer I get to this transition point, the higher the probability that I am going to see some citrate users evolve in this particular line. Right? So this is this type of an experiment now. So this is so again the benefit of having every I think they store samples every 500 generations. I think early on they were doing it much more frequently, but now the volume has just become so much. Now, because you have these freezer stocks every 500 generations, you go back and revive those freezer stocks, and you can replay this evolutionary experiment again. And every single time this happens, you plate it on a citrate plate, and you see what's happening. So again, what you're doing is you grow up cells, plate them on citrate, look at a plate whether there is anything or not, and so on and so forth, right? So let me just show you, just to give you a scale of the problem. There is a famous picture that these guys have of how many plates they had to check. All right. So Lenski, when he talks about this particular problem, he always shows this picture, and this is the picture. So this is the guy who did it, but look at the sort of the mountain of plates that are there, and essentially what is happening is he's just evolving these lines to a certain to some number of generations, and every once in a while he's just plating them to see if a citrate user has evolved or not. And by some estimates, the number of cells that were plated in this exercise were close to 40 trillion or something. So that's a huge screen. And what they see is that when they evolve any of these lines, the number of citrate users that they see is exactly 0. So these lines never get evolved the ability to use citrate. This particular line, the cit plus line, when it was evolved from a generation prior to evolution of citrate use, actually showed up 19 different 19 different times when the ability to use citrate was evolved. So if you're starting from scratch and or midway through, nothing happens, but only after 20,000 generations or so, cit plus appears exactly 19 times. Okay, so that shows the role of historical contingency. This is an interesting chart that they have from a paper that was published in 2012. And this is like what they show is that starting from generation 0, this is only for the line which evolved to use citrate. So what they saw when they did sequencing of the lines that what they saw when they sequenced what was happening in this culture was that this line had diversified into 3 different clades as they were evolving these strains. And you can see each one of these, I mean, this represents a certain mutation. And what had happened was that at this particular time, at 25,000 generations or so, these 3 clades were coexisting in the culture. And as we go forward in time, this clade disappears. This was eliminated from the population, and you had coexistence of these 2 clades. Around generation 30,000, you had citrate evolution, the evolution of the ability to utilize citrate, and all these red guys are marked as the citrate users. And from there on onwards, what has happened is that the first citrate users, all of these guys, their ability to use citrate is actually quite poor. They use citrate, but extremely badly. I mean, they're very slow at growing in citrate. Remember, this is an experiment where subculturing takes place every 24 hours. And growth on citrate only starts when all the glucose has been exhausted. So if you are very poor at growing in citrate, you just don't get enough time to grow on citrate. So their frequency in the culture actually grows very slowly. But by the time, so all these variants are getting eliminated from the population. They don't go beyond the first few thousand generations of growth on citrate. At this point, so we remember we talked about last time that many of these lines have evolved to elevate their mutation rate. In this particular, in the citrate clade, a mutator evolves about 3000 generations after the ability to use citrate evolved. And what this mutator did, now you're picking up mutations at a higher rate. And remember, the way we sort of understood the role of mutators was the following, that when you were extremely low on the fitness landscape, when there were lots of beneficial mutations available, you could presume that elevating your mutation rate increased the speed of adaptation in the culture. And something similar is happening here, that these individuals are extremely low on the citrate peak that we just saw, and this mutator evolved here, and that led to elimination of all these three coexisting variants. And since then, this mutator allele has evolved, and we have this new cit plus clade now, and this individual or this genotype is much better at utilizing citrate as compared to what it started with. So there is this population which is getting better at utilizing citrate. Interestingly, again, I'm not getting into the molecular details, but they are there in this paper. What has happened since is that as you are becoming better at citrate, there is a serious trade-off between how fit you can become at how good you are at utilizing glucose and how good you are at utilizing citrate. So as you become better at utilizing citrate, you become worse at utilizing glucose. So now this is happening in a sense that as these guys are increasing in fitness on the citrate peak, these guys are actually going down on the glucose peak. And these guys are unable to use citrate, but they are getting better at utilizing glucose. As a result of this, there is this coexistence between citrate user and a glucose user in this line. And this is an old data point of 40,000. These two are coexisting even now at 75,000 generations. The citrate user is continuously getting better at using citrate and continuously getting worse at utilizing glucose, whereas the glucose user has not evolved the ability to use citrate and is just getting better at using glucose. So, but what I find interesting is that when they evolved individual cells from these lines and looking at ability for CIT plus, when they performed this particular experiment, that they're replaying the evolutionary tape and looking for if CIT plus appears or not. When they do this from all these different clades, actually CIT plus ability arises from all three clades. So what they're doing here is, from this particular clade, they took 37 genotypes from this particular clade, and they evolved to check if CIT plus phenotype evolves in them. And out of the 37 trajectories that started from this particular clade, in 8 of them CIT plus CIT plus phenotype was observed. In this one, 97 starting genotypes were taken, and what's interesting is that it wasn't 0 here. 2 times here also you got the CIT plus ability evolved. And similarly here, that what that means is that the historical contingency actually started pretty deep into the history of this evolutionary line. Does that make sense? Questions? Okay. So what happened at a molecular level? again, the same paper gives these details. So what happened essentially was that in the evolved line, E. coli already has a citrate transporter. It's an antiporter, which antiporter is basically something that trades what is being brought in and out of the cell. So it brings in citrate and it throws out succinate. And now in the evolved line, what happened was that this region, and so the gene is called CYT, and the expression of CYT is driven by a promoter, and that promoter is only active in anaerobic conditions. If oxygen is around, that promoter is non-expressive, so that's 0. What happened in this line was that this entire region was just copied and a duplication event took place. So you have, it started from here, so part of CYTG was copied and going up to this gene called RNK, and this entire region was just duplicated and created another copy of this. And now what happened was that this city was driven by another promoter, and subsequent mutations in this promoter meant that this city was evolved in this city gene was evolved in conditions even when oxygen was present, and that allowed the cell to start using citrate very poorly, and then subsequent mutations took place which led to evolution of uh, citrate users, and essentially showing up as a phenotype as high turbidity in the culture. Because when this happened, the citrate users, they only noticed the citrate users when there was a phenotype, and the phenotype in their case was when they came to do the daily transfer, the culture was much more turbid than the other 11 lines. What they noticed later on was that the citrate ability had evolved 1,000 generations prior to when they first noticed the turbidity in the flask. And it was just that those citrate users were just so bad at utilizing citrate that you would hardly notice them in terms of increase in turbidity in the culture. All right. So what I find surprising is that why were there no mutations in the promoter region which would just enable this promoter to express citrate CYT gene uh, in oxic conditions, in the presence of oxygen, and especially because we know that every single nucleotide polymorphism has happened in this culture, but despite that it never sort of took off. And I wanted to juxtapose that with this paper that I really like that came out a couple of years back. So what these guys did was that we know, so this is the ancestral setup arrangement of the promoter and the gene of LAC. And LAC, remember, helps you utilize lactose when it hydrolyzes lactose into glucose and galactose. And this is the expression of LAC is driven by the LAC promoter. And this is the classical system that all of us have studied in whatever introductory biology class, where CRP and CAMP comes and binds at this promoter. And when no lactose is present, LAC-I comes and binds here and stops expression. But when lactose is present, lactose binds to that LAC-I, and this repression is relieved, and CAMP-CRP comes here and drives expression, and hence we get LAC-Z, and LAC-Z hydrolyzes lactose into glucose and galactose, and hence we get growth. So that is the natural arrangement. The question that they ask in this study is that how easy it is for a random sequence to evolve into a promoter. And to test this, what they do is that they replace the LAC promoter with a random sequence which is not a promoter, right, upstream of LAC. So now no transcription is going to take place because this random sequence is not a promoter. Does that make sense to everybody?
Yes.
So what you're doing is essentially you've just replaced the LAC promoter with a random sequence, and you don't when you do an experiment of this sort, you don't replace it with one random sequence, you replace it with many random sequences. Some of the random sequences might act in some sort as a promoter and drive expression, in which case the variant that you have created will grow on lactose. Whereas if this random sequence is not a promoter, does not drive expression at all, then you will not see any growth on lactose. And it's these sequences that you're interested in, the random sequences which do not drive expression at all. And then what you do is these random sequences which are not driving expression of LAC are evolved in a lactose environment. And in order to sustain growth, you add another carbon source which basically allows for some growth to happen, but not too much. So what they did was added a little bit of glycerol to the media. So the E. coli doesn't really like glycerols, but it does utilize glycerol for growth. So it does have ability to grow. But now if any mutation, what they are interested in is, the question is, what does it take for a random sequence to evolve into a promoter? And because there is lots of lactose, if any cell picks up a mutation here, that's a great way to enhance fitness in this particular environment. And because it's growth in lactose and everything else is already there, the lactose transporter is there, lactose metabolic genes, everything is there. The only thing is, even LAC gene is there. The only thing is you need to express this LAC. And for that to happen, this random sequence needs to acquire mutations such that it becomes promoter of some sort. And the stronger the promoter, the better the growth on lactose is going to be. And what they find is that if you take any random sequence, it only needs one or maximum two mutations for this random sequence to start acting as a promoter region. So promoter regions have a characteristic architecture in the sense that transcription is done by RNA polymerase, and all these promoter regions do is recruit RNA polymerase and start transcription. And RNA polymerases have specific binding site on a promoter, and they bind at these sites on promoter and thereafter start transcription. And what they find is that if you were to take any random sequence, it doesn't take a whole lot of mutations, in fact only one or two, such that this sequence can resemble, there will be some element in this random sequence which looks like the site at which RNA polymerase will come and bind. So promoter evolution is actually fairly rapid, is what what the study shows. In general, let me ask you this, that what is easier, quote unquote, what is easier to evolve? Expression of control of regulation, up regulating or down regulating something, or changing enzyme function? Does the question make sense?
Yes, sir.
So is it easier for evolution via acquisition of mutations to enhance an enzyme's catalytic activity, or is it easier to suppose we needed more of an enzyme? Suppose we had an enzyme amount in the cell which is catalyzing 50 substrate molecules every second, and that's not enough. The cell can enhance its fitness if it was catalyzing 100 substrates per second. Now it wants to catalyze more, but this can be done in two ways. The first way is that you just make more of enzyme with the same activity. More enzyme means more catalysis, and hence you move towards catalyzing 100 substrate molecules per second. The second one is you produce the same amount of enzyme, but just more efficient. So instead of each enzyme molecule catalyzing 10 substrates per second, it's doing 20 substrates per second, and hence you get to 100 substrates per second at a cellular level. Which of the two mutations do you think is easier or arrived at earlier in an evolutionary experiment? Anything?
I think efficient enzyme production arrived earlier.
Okay, why do you say that?
Because
It could be a gut feeling also. That's
yeah,
okay too. Yeah.
not really a particular reason.
Hmm.
So the second one you said is changing the enzyme.
Hmm. Yes.
Not change. I mean, not exactly changing the enzyme entirely, right? It's just efficiency of the enzyme is improved.
Yeah. No. I don't think this actually means changing the enzyme as in you bring in a new enzyme. It's just improving the current enzyme.
Yeah.
Others?
So I guess it will depend upon like if it is a functional group, that enzyme, and if it can be easily, but I guess it will be totally upon chance, because even if we want to increase the copies of the gene coding the enzymes, it will be again a chance event.
No, of course, everything is a chance event. Okay. Others?
I think it'd be easy to change the expression levels.
Why do you say that?
Well, I'm not sure. Based on the experiment, at least, it shows that it only needs one or two mutations.
Yeah, sure.
And most of the mutations in enzyme in the enzymatic gene itself might
Yeah.
be deleterious or neutral. So you might it based on this experiment, at least, I think it'll be easier for gene expression. And usually, I think that's what most of the eukaryotic species also do, in the sense that there is more regulation in expression rather than they heavily utilize this gene expression property rather than
Yeah.
changing. So that I think that's why.
Yeah. No, that's true. So this is a lesson that we know from not just this experiment, not LTEE, but a lot of evolution experiments, that it's much easy that regulation evolves over a much shorter time scale compared to enzyme functionality. It's much quote unquote easier for the cell to upregulate, downregulate via transcription, expression of enzymes, rather than changing enzyme uh, capacities. So these things happen on a different order of time scale. So transcription evolves much more faster as compared to catalytic activities of the cell. So in that sense, I do consider it a little surprising that why did it have to involve a whole duplication event, and then SIT-T gene to be driven by another promoter, and then subsequent mutations to take place, and then citrate phenotype to evolve. Why could the SIT-T promoter not pick up mutations such that the repression in oxic conditions? So what is happening is that in oxic conditions, the cellular regulatory arrangement is wired such that transcription doesn't take place, and hence you have a repressor that comes and binds on the promoter. But any mutation, and we know all mutations have taken place, any mutation that took place, that takes place, that relieves this repression event, uh, is going to lead to expression of SIT and hence citrate utilization. So why it does not happen is an open question. But some of the things are just so hardwired in an organism's regulatory network that they are just very difficult to get rid of. For example, glucose repression is one thing. Yeast, for instance, has evolved over millions of years, and yeast just loves glucose as a carbon source. So what happens is that there is a repression of a large number of genes when glucose is around, because it knows that glucose is around, and hence it does not express any other, it does not express genes which are responsible for other carbon source utilization. Let's say galactose, for instance. So when glucose is around, the galactose genes are just off in the environment, and this glucose repression is so hardwired over millions of years that it's just very difficult to evolve a strain where this glucose repression is eliminated. So some of the regulatory patterns inside a cell are just not easy to get rid of, and perhaps this is one of them. That it's just not easy to get rid of the oxic repression in the CYP promoter, and hence it needs a very different molecular route to evolve citrate utilization in oxic conditions. Okay, all right.
So,
Yeah, go ahead.
yeah, so just a thought. If there wasn't a study, right, where they studied the ancestral promoter and the new promoter, the
Yes.
linking mutations required for this change. Means they
Yes.
started with a random sequence and then went to a new promoter, right?
I'm sorry, are you talking about this last
The lag
slide?
C.
Yeah.
Lag C.
Yes.
So maybe if they studied the mutations from the ancestral LAGC promoter to the new LAGC promoter, they might stumble upon a special mutation which cannot be achieved due to fitness uh, valley.
No, I'm sorry, I didn't understand. So you're talking from the perspective of this paper, right?
Right.
So I didn't exactly understand what are you saying. Say it
So I'm
again.
saying the ancestral LAGC promoter, which suppresses lactose expression.
It doesn't suppress
Oh,
lactose.
sorry. I mean, the original LAGC promoter, we are replacing it with a random sequence. So
Yes.
instead of replacing it with a random sequence, we already know the new promoter. And if we see the mutations required for going from the ancestral promoter to the new promoter, maybe in these mutation pathways, we can come across one which, I'm saying that in these mutational pathways, the organism should pass through a valley, fitness valley, and that's why the mutation couldn't be achieved, right?
Let me say, I think I didn't understand, I didn't explain the experiment properly. So this is the ancestral promoter, the LAGC promoter, right? You replace this with a random sequence which is not a promoter. So let's say this is the ancestral promoter was something, and you replace it with 100 A's in a continuous stretch, right?
Okay.
Now 100 A's is not a promoter. Okay, so RNA polymerase will not come and bind here. Now the question that I'm asking is that I have 100 A's, and downstream of 100 A's I have LAGC, right? Now I am going to evolve this particular strain in a lactose environment with the slightest bit of glycerol environment so that there is growth. And the question I am interested in, that what does it take for a random sequence like 100 A's to become a promoter?
Right, yeah.
Right? So what I'm going to, so maybe if I have a G here and a C here, if I have a G at a 30th position and a C at a 50th position, that sequence acts as a promoter. So I need 2 mutations for this random sequence to turn into a promoter, right? So I evolve this starting sequence, 100 A's and LAGC. I evolve it, and I can always monitor when there is growth on lactose, right? I can simply tell. And when that happens, I am going to get this promoter region sequenced, and see how many changes did this promoter sequence need. How many changes did this random sequence need in order to become a promoter, right? So in that sense, I am comparing. So this promoter has evolved via acquisition of mutation starting from this random sequence. Does that make sense?
Yeah, I got that.
So मुझे question नहीं समझ आ रहा.
I got the experiment. It is means above this experiment. Apart from it, now we know this new promoter which was they have developed, right? So if we compare the ancestral
Not they have
promoter,
developed, which has evolved, you mean?
Yeah, which has evolved, correct.
Correct.
So if we compare the ancestral promoter and this evolved promoter,
yes,
we know the genetic changes required.
Yes.
And we can.
No, but that's too many. That's too many.
Yeah, okay,
that's too many. We still don't know that what sort of mutations are required in the original promoter to make it sit plus in oxic conditions. We don't know that. Okay. So typically what happens is, okay, Anirudh, let me just make this point. Anirudh, you come in then. Typically what happens is that if you have a promoter and there is a repression event going on, so LACI is repressing lactose expression, and oxygen has many regulators, so OxyR is one. What these repressors will do is they typically recognize a binding site, and that binding site will be 10 to 15 bases, and they will recognize that site and come and bind here, and their binding would just occlude RNA polymerase from coming and binding on this promoter, right? So, and the nature of binding of each transcription factor is unique, and some transcription factors are very specific about the sequence they are binding. So if they bind 15 bases, if it's 13 bases they will bind, but if it's 12 bases they will not bind. But some are more promiscuous, they will bind even if if 10 of the 15 are there, they will bind. So the number of mutations required to get rid of that repression event might be dependent on the nature of the transcription factor that's controlling expression also. Yeah, Anirudh, go ahead.
Yeah, so one of the previous questions you asked was why does it take a complete duplication for to use citrate, right?
Yes.
But I mean, as far as what you've taught us, you mentioned that so this only occurs. So this whole gene transcription of the citrate transporter only occurs when you don't have oxygen. Yes. And it seems that if it evolved into a promoter that would be expressed even during when you do have oxygen, and then at that point, wouldn't it change the energy utilization of the bacteria? Wouldn't it unnecessarily have to create citrate transporters? So maybe that could be the reason why the selection. It wasn't an efficient way to do this.
No, it is an inefficient way to do. It would be, but I think the gains in this particular environmental context are just so high that if we were to place a mutant version, suppose we created a mutant version which was using citrate in oxic conditions in competition with the ancestral strain in the environment in which this experiment is being done, I don't think the mutant that we place is going to get competed out of the culture, because the gains are just so much. If you look at the exact recipe of how much glucose and citrate is there is just a lot of glucose, a lot of citrate in that media. So and the energy cost of a particular protein in the worst of cases is not going to be more than 1 or 2% of the energy budget of the cell. It's not going to be more than that. So in this context, I don't think that's what would have happened. Does that make sense?
Okay.
All right. Last point, which I really like this. I don't know. So So I want somebody to explain what happened in these two functions. So remember the two papers that I had shared? One was the citrate one was this paper in Science in 2016, something like that. I want somebody to explain what happened here. Somebody.
So one is a power law, the other one is a hyperbolic law, and
Okay.
and what it kind of says is in the hyperbolic law.
No, tell me what they did.
Oh, okay. So they used, so they modeled the number of, so they compared the average fitness, mean fitness over the number of generations, over the number of generations, and they modeled both the hyperbolic law as well as the power law with the experimental data. And they found that the power laws seemed to be more fitting.
Yes, but you're missing an important link there. How many of you did not look at the paper? Please raise your hands shamelessly. Okay. Okay. All right. Okay, fine. So here is what they did. So Anirudh, there is an important thing that you miss in your explanation. What happens is, so this, let's consider that this point is 20,000 generations. If this is 20,000 generations, then remember they are storing every 500 generations, they are storing samples. So every 500 generations, they have data point for mean fitness of the population. So there are 40 data points in this window from generation equal to 0 to generation equal to 20,000. And obviously it's going to increase and the trend is going to look like this. And let's say fitness is expressed in relative terms and F naught is equal to 1, which is the mean fitness of the ancestor. This is just normalized to 1. And in 20,000 generations, I think they see something of the order of, I'm making up stuff here a little bit, but maybe 1.6, 1.8. So there's definitely more than 50% improvement that has taken place in 20,000 generations. Now, with these 40 data points, they have fit two functions to capture this dynamics of increase in fitness with number of generations. And G is the number of generations. And in the first modeling exercise, the function that they used is this, that mean fitness of the population as a function of number of generations is simply F naught, the starting fitness, plus AG divided by G plus B. G is the number of generations, A, B are variables to be found out. And they find A and B which best fit the experimental data. The second function that they use is Fg equals to Ag plus F naught, which is just 1 raised to power B. And A, B are again constants to be found which best fit this data. So using the data from 20,000 generations, they find A and B with respect to each of these two functions. So now we have two predictors of fitness as generations go by. The reason they chose these two different representations is because there is a very important difference in what these mean mathematically. The first function has an asymptote. This function will always be less than f0 plus a, because a and b are positive. So when g reaches infinity, this term in the bracket simply becomes a. So the fg is always going to be less than f0 plus a. In infinite time, this becomes f0 plus a. So this is a horizontal asymptote for this function. On the other hand, this particular representation has no asymptote. There is no peak for this function. This essentially can reach any fitness level given long enough time. So after fitting these two to the data from 20,000 generations, the degree of fit, and I forget the exact numbers, the degree of fit associated with these two representations is extremely high. I mean, its R square is of the order of 0.99 or something. So for each one of them. So both are excellent predictors of the trend that is observed. But then what they do is, of course, they have data for several more generations. So now they do the same exercise. So now I'm changing the time scale, and now 20,000 is here. So this fitting was done with this data in mind, and now they ask that which one of the two is a better predictor of what happens in the future, and now this point is 40,000. So the fitting was done with the data from the first 20,000 generations, and now they are asking that which. So up until here, both fits are excellent because these are fits using this data. But now they are using these two functions to extrapolate the fitness trajectory from 20,000 to 40,000 and asking that which one is a better predictor of what happens in the subsequent 20,000 generations. And Anirudh, does that make sense? That's the important difference.
Yeah.
Right? And what do they see?
So in that case, I think they saw the power law making more sense.
Right. This came out to be a much better predictor as compared to this one in the time of 20,000 to 40,000 generations. 0 to 20,000, both are excellent, but in the subsequent 20,000 generations, this comes out to be a much better fit. What do you think that means, that the second representation is a much accurate predictor of what happens in the evolutionary trajectory than the first one? Is that good, surprising, bad, not surprising, terrible? What does it mean?
Doesn't it go against the idea of peaks in that case? Because
that's why it's a science paper.
Yeah. So in the hyperbolic case, there's an asymptote, so that would mean that you've reached the peak or something like that.
Yes.
But it seems in the other power law, it's always increasing no matter what.
Yes, exactly. There is no peak. Is that idea uncomfortable? Others, I mean, those of you especially those of you who have not read it, do you understand what is being said in the paper?
Yes, sir.
Others?
So for the given number of generations, this model is a good predictor, but how efficient this is for further generations, like large number of generations?
No, who knows? I mean, sure. So this turns out to be a better predictor for 40,000 generations, but we don't know if it's a better predictor for 400,000 generations. I know that at least till 70,000 generation, when you repeat I mean, now the data is available and you already have the A and B's associated with the two models, so you can yourself check. Up until 70,000 generations, the second model is a better representation of what is actually happening. But beyond that, it's anybody's guess.
Sir, doesn't this talk about the shifting maxima as well?
Yeah, sure. Absolutely, sure. So what that means is that while you may be keeping the environment constant, that doesn't mean, see, landscape is defined, landscape is a relationship between sequence space and in a particular and the particular environment. Now, while we might be keeping the environment the same, although that's debatable also, and I'll come to that in just a second, but sequence space itself is a variable quantity. It's not as if that I am keeping the sequence confined to L. As we just saw, then one of the most dramatic transitions in the evolutionary trajectory happened via a duplication event of a small region, 1,000 bases or so. Now that 1,000 bases immediately changes L. And if L changes, the entire sequence space associated with the culture, with the organism changes. And hence that's going to change the context in which the landscape is viewed. The second reason is that the rate at which an organism is growing, or the metabolic capacity of the organism, is actually also changing the environment in which this is taking place. If you had slow growers in cells initially, then the environment in which this experiment was taking place was that glucose was being exhausted slowly, products like acetate were secreted slowly, so the environment had a particular dynamics associated with it. But as fitness is changing, the environmental transitions taking place, their dynamics have also changed. Metabolic waste products are being secreted at a different rate, they're accumulating at different rate, and hence they provide hindrances and opportunities to the organisms in the environment at a different rate. So in that context, the environment and the genotype are both changing, and hence landscape is a very dynamic quantity in this context. So it's not a static structure at all. As regards to, I mean, one of the major discomforts is that what the power law prediction being more accurate suggests is that the organism is just going to get fitter and fitter and fitter. I mean, there seems to be just no cap on it. And if you plug in, how fast does E. coli divide in any environmental context? Anybody?
20 minutes.
20 minutes. When everything is given to it, rich media, it divides in 20 minutes or 37 degrees. Experiment also is also at 37 degrees. It's 20 minutes, right? This predictor, the power law predictor, says that E. coli will divide in, again, I did this calculation a while back, but I could be a little bit off. It divides in like 8 minutes in 2.5 billion generations. Okay, so while there is no cap associated with it, would you say that E. coli dividing in 6.5 minutes, I'm sorry, I don't know where 6.5 came from, but E. coli dividing in 8 minutes or so in 2.5 billion generations from today is inconceivable?
No.
Others?
But I think it's possible.
I think it's possible too. Right? I mean, essentially, we have 2.5 billion generations to go, because what we have done is zero, right? 75,000 is nothing. So I don't think it's inconceivable at all. And hence, I think the power law predictor is not bad at all, because while it may suggest that there is no cap, but also the A and B are such that it's extremely flat, right? And the amount of time, it's just an eternity, really. And as institutional an experiment as LTEE has become, I really don't think that there is any guarantee that the experiment is going to go to 2.5 billion. I mean, who knows? I mean, it's not going to go to 2.5 billion generations.
But sir,
Yeah.
There's the question of in vivo versus in vitro as well, right?
What does that mean?
I mean, in its natural environment, may this kind of behavior might not be seen.
Yes,
Just because we're performing it in a lab, we came across it.
absolutely, absolutely. No. So that brings us to the, see, this is the theme that we started with at the beginning of the discussion on experimental evolution, right? That the ecologist's critique of studies of this sort is going to be uh, that this is not representative of how evolution takes place in any ecological niche, right? There are species interacting, environments are dynamic in nature. You don't get this sort of consistency in any environmental niche at all. So this is not representative of how evolution will proceed in an environmental context. And increasingly, as things are getting more detailed, what somebody will argue is that look, under, if I perform an experiment under this specific condition, I see this particular phenotype which confers 3% advantage to this cell compared to this other variant, and hence this is an important mutation. While that is true, and when is that spandrel submission due?
7th April, I guess.
7th April. So I guess you not started looking at it. But can we hold this discussion till we have all read that paper? Okay?
Yeah.
So essentially, the argument is that somebody will argue that variation just exists, and somebody will try to find some environmental context in which that variation is advantageous, and hence say that look, this variation arose because it's advantageous, and hence selection is important for the evolution of this variation. So the spandrel paper is a critique of people who try to find an adaptationist explanation for every sort of variation that we see out there. But we'll come back to that in 10 days time when you've all read the paper. Anirudh?
If you use a species that has a much higher mutation rate, for example, then you could see it reducing its increasing its fitness in a much faster pace.
Yes.
Right?
Yes.
But isn't it scary that it's possible that at some point, if the species is highly contagious or it's pathogenic, then and it also has a high mutation rate, it could essentially change from 20 minutes of division per division to 3 minutes per division, and that's kind of, it just feels, that seems 8.5 is still, I don't know, it just seems like 3, 2, 1, they seem way worse, especially if it's pathogenic.
Yeah.
But I don't think people have seen that though, right?
Yeah, no, I don't think so. But also, see, in all of lab experiments, there's an important distinction here. In all of lab experiments, what is fitness equal to? What do we assume fitness equal to?
Number of progeny an organism able to create.
Yeah, but in a prokaryotic setting,
The replication time.
replication time.
Okay, yeah.
Yeah, right. Inverse of time you take for division. But that need not be a very accurate measurement of fitness in an ecological niche, right? Because if it's not just faster replication, right? Often fitness in an ecological niche is will be defined by your ability to just not die, and not just because there are going to be periods when there are no resources, you're struggling for them. So your fitness is essentially just not dying instead of replicating as fast as you can. Does that make sense?
But I mean, if we consider doubling time, it pretty much means the same thing, right?
No. So imagine a strain which divides extremely fast when glucose is around, but in the stationary phase, its survival is really bad, right? And compare that with a strain which is growing at much slower pace in glucose, but it's extremely robust at handling periods of starvation, right? Then if you place these strains in an ecological niche, in that particular environment, the fast grower is going to lose.
Right, I hadn't considered that.
Right. So that is another big critique of work like this, that this sort of work attributes fitness as just equal to inverse of the division time associated with a particular genotype, and it's only done for the purposes of convenience, because that is the easiest thing that we have to measure. But in an ecological niche, that the definition of fitness that is being used in this context is not translatable to an ecological niche. But even without that, the point you make about high mutation rate creating havoc is already doing that, right? I mean, you see that playing right now.
Yeah.
Right. So I mean, that's the reason that viruses. Why do you have to get flu shots every year? Because every year there is a mutation, and the previous year's shots are not going to work, and that's a problem. Right? All right. So we can continue this discussion if you guys want of there being no adaptive peak if the idea doesn't sit very comfortably. It doesn't sit very comfortably with me, but I think it shouldn't sit comfortably with anybody, and that's okay. Let's stop for today. One thing, our exam is a viva, is a one-on-one viva, and I would like to do that in the week of 20 to 26 when there are no finals. I think finals start on 26th. So if it's okay with everybody, last day of class is 20th, which is a Tuesday. So for us, it's going to be like 16, 17 or something. So I will share a schedule when where I'll like to talk to each one of you individually for half an hour or so, and that will be the final. And I'll do that in the period from 21st to 25th or something like that. Okay. Please don't tell me that you have to study for the Viva. We will just chat about different things in the course, and you don't have to listen to all the lectures that we've gone through. Is that okay, everybody?
Yes.
I am not really giving you much of a choice, but I'm okay with that. But seriously, do not think. I mean, I personally think that almost everybody has been pretty diligent in what we've been talking about. So there is absolutely no need for you to go to lecture 1 and watch the videos at 2x speed and come to the Viva. Just show up. We'll talk it. And as you have seen, there aren't many right or wrong answers in evolution anyway. And the course has been a very broad introduction to a subject which is extremely large, and each of the sections that we've talked about could easily be an independent course in itself. So we just scratched the surface of many things, and yeah, please don't feel obliged to watch videos and come. So I'll share the schedule with all of that. And the second thing is, would you like any more assignments to be given in the course, or is what we have done enough? This is your chance. You have to say no. Akshat.
हाँ.
What do you mean, हाँ?
I mean, मैं क्या बोलूँ? It's popular opinion.
And you get to be the leader of the popular opinion, whatever you say.
Oh
No,
my
no,
God.
I don't know. I mean, are you guys overwhelmed with assignments and stuff like that? People are not reading papers. So, if you are overwhelmed at this end of semester, I understand. What do you personally think?
एक दे दो और but like with an extended deadline, like अभी दे दो और बोलो कि अप्रैल एंड तक सबमिट करो.
Chalega. And we'll make it optional. Okay, I will consider n-1 assignments out of n.
हाँ, perfect.
Okay, cool.
N-2 भी चलेगा.
1 is okay. Just don't push your luck.
By the way, the midsem counts as assignment or a separate midsem?
I mean, to be really honest here, I don't even. I mean, as I said, no, that I think everybody has tried hard in the course, and to be really honest, that's all I care about. So let's not worry about grading. I'll tell you what your grades are before submission, and we'll visit that problem then.
Okay. Just a doubt. Before closing. Generally, the microorganisms are in which phase of their growth in their natural habitat?
Stationary.
Stationary, right?
Yeah.
I thought so.
Stationary, and if you're talking of microorganisms on surfaces, which a lot of microorganisms are, then they are in their. It is also stationary, but they form up these particular structures called biofilms.
Okay. So that might give us a lead why we don't see fitter.
Yeah, sure, absolutely.
Okay, thank you.
All right, I will see you guys on Wednesday then. Bye, everybody.
So the paper for the next assignment, it had restricted access. So
I will share it. What are we talking about? Which one is it?
the spandrel one.
Oh, really? Okay, sure. I will share the PDFs.
Okay, yeah. Thank you.
No problem. Bye.
Thank you, sir.